who's around on this, but going to talk about biophysics. And I guess this is something which you guys might not have been exposed to. So my goal was to do a broad intro first, and then we can go into some specific problems that I've thought about a little bit more. Good? Sounds like a plan? And again, just interrupt whenever, okay? 4 people. So, you know, do not use the most of it, okay? You've shown up, so at least ask questions. Okay. So I just also wanted to say I have a very sort of weird background to be a physicist in some way. I started in electrical engineering, then worked for a couple of years, then did bioengineering, then did physics, and then I did my PhD at Princeton, then I was a postdoc in theoretical physics in Santa Barbara, and then I ended up here, and now I'm doing experiments and sort of theory both, right? So it's kind of weird mishmash of things, so that kind of shows the kind of problems I find interesting also. All right. So I just wanted to make this point up front that, you know, biological systems are really what we think complex, right? In the sense they have lots of moving parts, right? Even to understand, I mean, a cell is already pretty complicated, but let's not think about from the molecular scale. Let's say you want to think about walk today, right? Walk. So people still are trying to understand how do we actually manage to stand up and walk, right? What are the balances? How do the joints work? And that's the problem in robotics to solve. It's just being solved now, I guess. We may know more about it, right? So it's we don't have good models even for sort of simple phenomena, right? And then the phenomena can get complicated, and then we get kind of even a little bit more difficulty because that's some things we have talked in the class. So that's sort of what we mean by complex, right? A very specific meaning in the sense. The goal is to try and find sort of, when I say simple, I'm going to try to clarify what this simple means. The idea is that we can't explain everything, right? The goal is not to take any single or any phenomena you take. Right? Can I explain all leaves in the world and their shapes? Not. Maybe it's too hard. Some man to do it. But it feels like a complex and a fast. But they came up with a very simple model. Okay, maybe I'll talk about it instead, right? So what they found was you can take any leaf in the world, pretty much. I think there's distinction between 2 types. Once you fold it, there's always a fold for leaf so that all the edges of those leaves are straight line. Sort of like an origami one. You can fold something, then you make one cut to get basically shape of it. And it goes back to the development of leaves when they come out. If you actually now start observing these sort of little pods where the leaves are going to come out, you can actually see all the edges on the top. And when it opens up, it kind of basically opens up in this funny way, right? So in some ways, that's a very simple model of this complicated shape. Does that make sense? Right? So that's the goal of what we want to do in physics. Right? A biologist might be interested in a very different question, right? So it also matters what the questions ask. The system is the same, different people can ask different questions, and that's what sort of distinguishes a little bit as a physicist asking a question in biology and a biologist inquiring about the same stuff. Right? Not to say, you know, there's no judgment call on either of them, it's just a different way of approaching the problem, and, you know, what I'll try to make a case for is that sometimes these approaches can also, the kind of approaches we take, can also yield interesting insights into the system. So before taking some very specific examples, I wanted to give you really broad spectrum sort of view of biophysics, right? What are the kinds of problems in biophysics, if you will, right, at different scales. So I'm going to start with some videos. We can give a little more insight than words. And you guys have seen this video. Just watch it. What you're watching is a big cell. Okay, let me just pause it so I can kind of orient you a little bit. So this giant cell, if you will, is a macrophage. This is one of our immune cells, right, which is responsible for a lot of immune response, first immune response that we get. Either when we get an infection or we get a cut or something, you know, when lots of bacteria are starting to go, these guys are sort of come and clean up. So, and is it a stick or something? Point? No. Maybe just stick in these rooms. Okay, never mind. Right? So you see this little sort of bacteria. This thing is chasing this. Right? That's what's happening. So let's see what you see. Bugs are moving around. Guy keeps chasing. You sort of see it has brain of itself, you know, it has to make decisions what to do, and gets it, right? So there's a lot of decision making going on here, right? Which directions to go, where it might be, how fast to go, what that is happening at a single cell level, right? This is what we do when we're chasing something, right? Go left, go right, how far it is, change the speed. So all things that we think our brain can do, a single cell can do pretty well. This started a whole host of questions of how even single compute, they do computation, and then they have to say something, compute something, and then actuate, also have to do think about it. So I think this is a slightly older video, and this kind of took a lot of work about physics, this kind of work. Cell. Good. So you can think of inside, they're talking to each other and deciding there might be a protein receptor site picking up a signal, and that signal transduced side decides, how it is to do what is atomized sort of filaments, which like the structure components, and you have to create in turn this object. And this has to happen at very fast time scale. So this is sort of at the cell level. Now you understand the need to go with the cell to understand what's happening like this. This is sort of one. Now I'm going to go to the slightly bigger level, the multicellular organism. And you look at a very early stage of it, embryonic stage, if you will. So this is a fruit fly, these flies that float around. That's become very interesting model to understand development, also other. So this very early stage is egg, which is fertilized. Each dot that you see is actually a nucleus, not the cell. So what happens is they make a lot of nuclei without even making cell membranes. You're going to see a video, and then we can talk about it. So this is very early stage. One more round. So what is happening? Any guesses? Right, so they're just dividing. I'm going to pause it here. Right, so this is like mitosis, right? Each thing is sort of just dividing into two. The amazing thing is the whole thing works in a very coordinated way, right? Physical systems don't coordinate that well. You have to work very hard for things to be coordinated, right? Otherwise they would get uncoordinated.
The coordination is in the kind of the wave-like.
Yeah, exactly. So this wave type sort of kind of wave goes through, right? What establishes that wave? Where is it coming from? Why is it important? All sorts of questions then open up when you see a phenomena like this, right? And you can think of it as a dynamical system, waves propagating through. But then you have to figure out where they're being generated, and henceforth. So that's another class of questions which people have been worrying about another last 7-10 years, trying to think about what is happening. And remember, this is a fruit fly that has to figure out pretty quickly which side is head and which side is tail. You can't look at it and tell, right? And then it has decision makes to be done. And it needs to, the question is, does it have that information? Does it not have that information? Is it given by the mother? Does the cells figure it out themselves? How precise does it have to be, right? If you take a lot of Drosophila, do they all get the same answer more or less within what kind of errors are there? And those errors are starting here, right? So if some of these nuclei move around a little bit and you're going to get a different place where the wing is placed, what is happening, right? So the idea of, how do you kind of figure that out becomes extremely crucial. So if you take this thing, let's think of a very physical system, and put it at a higher temperature. Just take this egg, put it in a hot plate. Not hot enough to burn it. What do you expect to happen? Well, we're not heating it to level it cooks, right? I know it's getting to dinner time, but so what happens is keep the wave, it divides, right? The rate of division comes fast, right? Something responds to temperature, but something doesn't. Right? If they were really little particles, they would actually start yielding radiation. So you don't see that. Let's up it a little bit. Let's say I only up half the egg. Do you think that will happen? Yeah. Faster. Seriously, what happens is the hot parts start dividing faster. If for sure this is not going to use a good fire, let's get realistic. Yeah. Yeah. You keep it great. Right? The thing is, it's hard experiment. Small thing, you kind of keep it. Now, quick, the opposite temperature. It's a hard experiment. But there's something really interesting about biology. What happens is one has dividing faster. At some reason, it basically waits. Things stop doing. They wait for outcome. Then will they push again within speed. So there are these funny checkpoints in development which allows something to actually develop into a fly. Right? Actually, don't know the answer to that problem. Right? And this is an experiment which many biologists wouldn't think of doing. Right? The guy who did this experiment, but it was really very recently. In the scheme of things, people have been flying embryos for right? Goes back to establish things. So that is only that as from physics, engineering, math perspective, you can idea to analyze, look at it. You can even think of crazy experiments which other people might not, because you have to see that things when the temperature is high. Well, it work, right? And you discover a whole new now, which is that's sort of another equal. All these beautiful that you see in biology, all these sort of things that you see in inanimate stuff, if you will, comes from where? That's this idea they have. How did they all come about? Darwin, right? So things have evolved. They have evolved into something because this is better. Whatever from there, right? It's tall neck of the giraffe or something like that. Right? It's because useful. So how to study evolution? It's a very hard problem. Do the experiment drafts, it's long, etc., etc. Although bacteria provide a good way of looking at stuff. So bacteria is another single-cell organism. It divides, and existence is a classic example of evolution happening in front of our eyes. These bugs will not take a population of bacteria, might not antibiotic resistance, in the sense that if I give an antibiotic, they all die. They can evolve antibiotic resistance. And the way to do it is they mutate and they selection. Right? So this is a fantastic video. We kind of made this very visual. It also has audio actually. Maybe I'll try to play it. You hear it?
Except they're 9 bands. There's barely more than the equal life survive. The side of that's 10 times as much, 100 times, and finally the middle has 1,000 times as antibiotic. Then across the top of it, or thin agar, that bacteria move around it. The back is black because of it, and the bacteria here is white. First, see they spread in the area of the antibiotic until they can longer survive. Then a mute appears on the right. Resistant to the antibiotic spreads until it compete with things around it. When they hit the next boundary, they have to pause and develop mutations to make 10 times as antibiotic. And you see the different mutants repeat this one. And after about 11 days, they finally make it into 1,000 times as antibiotic and wild type survive. And so we can see by this process of accumulating successive mutations that bacteria which are normally sensitive to an antibiotic can evolve resistance to extremely high concentrations in a short period of time.
Familiar with it? You can try to give your hand. It's not connected.
Yeah?
Yeah.
Okay. All right. So,
time to survive. And so it involves this
so each of these sort of events that these guys are putting out, the here example, this is administered by a single cell, right? So what the Darwinian is, have a lot of cells with no antibiotic zone, but they are growing, they were circulating mutations. Some had some arbitrary, right now mutation doesn't do anything. It makes them a little better, if it, right, growth. But once the antibiotic comes, these mutations give them the benefit to grow and stuff. And it just happens that there was a handful of mutants at its front edge, which kind of had somehow discovered this mutation and gave them an adjuvant antibiotic growth. Right? And that's purely chance. Right? It's not happening after antibiotic came. They were already there, the antibiotic showed. And this can only happen when the number is really large. Right? The number of bacteria, I don't know, 10 or something like that. And each one mutating at once or twice every generation. It's even a lot of mutant mutation kind of happen. Right? And this sort of material of evolution. If there's no antibiotics, this antibiotic is bacteria. It's just not growing as for the other ones. Right? So if you pick up piece of dirt outside, you know, actually tons of resistant bacteria are also antibiotics. Randomly bred, right? Just not selected. So why do we have lots of antibiotic resistance in hospitals? Because there are people with antibiotics there. Right? So while they're here, you didn't have any benefit in outgrowing the regular cells. Once you have antibiotic, they have a chance of becoming more dominant. This is part of evolution, right? This is what has resulted in the macrophages that hunt cells as guys to this little flag which can figure out appendages and that. And as physicists or as scientists, what we're trying to figure out is the root of these things. Right? What has evolution itself? And now you should not be surprised that there might be many possibilities. Right?
So we end up building
I don't just want to touch it every minute. Someone touch it. And the other happened. I did talk like 3 days ago and it was perfectly fine. But hopefully not my computer. Yeah. Okay. Well, try to make it. All right. Questions?
So you're saying that it's because the mutation happens that's just not as special, if it's like not that high biology, it's possible that one of the bacteria of the other phage has a mutant that can be pushed forward to all the
100%. Yeah, absolutely.
but it's not selective, and they're competing with all the other enzymes.
Exactly. Right. That's one way of thinking about it. The other could be to get to somewhere at 1,000-fold concentrations. Maybe you have solved a simple problem first. And maybe you need 5 mutations. Right. Imagine to get to antibiotic resistance of 1,000x. Right. Getting 5 mutations is less improbable than 1. Right. Because essentially the independent events. If the probability of getting them is p, getting 5 of them in one generation is 5. Right. P is less than 1. It crashes out. Right. So that's another aspect of it. Right? For example, famously, you constantly generate tumors. Right? All the time. Now, our immune system clears it. Eventually, some cells get cleared up and get more than 1 or 2 mutations. That allows cancer to then escape and progress. Right? So there is this sequentiality also to evolution. Right? And this is when you have biotic resistant bacteria, you actually give cocktails of drugs. Right? Cocktails sort of kind of shield from different directions. Right? So you only had one direction, they mutated one or the other. You sort of, like especially for HIV, it's a very rapidly evolving virus. You need to have skills that are designed so you can check in various directions we have found out that kind of. Good?
The mini video is like a whole area of the bacteria. If one of those is not on the have mutation, does it move there? This is
this is not because kind of very like a biophysics, it's sort of not to grow out but run it, right? So it's like a film of area, not remove much. They basically grow and they push birds and so think of them just in front. That's what it is.
So one is on
one of them has to be a boundary. 100%. Absolutely. The full problem for stochastic dynamics. And that's how we try to do about it. And again, stochastic dynamics wouldn't tell which way you're going to mutate. But you ask questions, chance of getting even a mutation. Right? Many do it is a question you can ask from calculation, not which. Right? How the question would be different. Biologists might just which gene mutated or not biologists. The fantastic question. You cannot calculate or whatever that question, right? That's an experimental question, right? You can still do interesting experiments like these, which maybe a biologist might not be interested, and that might teach you something else. Okay, let's come back. So I still have 20 minutes.
Yeah,
Give and take. Okay.
maybe a little more.
Main
but-
point was to introduce you to things. Don't have to talk about do. Yeah. Going back to that cartoon I just so the goal, I said, was complicated beast from life. We're trying to find some solution. Right? It's not simple. It's up to you if the idea you want to find a goal. Right? Is that a good decision, or you want to agree? That question asked, but mostly our goal is to this part of the world. Right? It's a very complex problem. We want to find the bare bones essential to understand. Whatever aspect you want to understand. Right? I'll tell you a very simple cartoon. Something. This is something I found many ago. This came from an experiment. We're looking at a monkey. This is not, but as a model for what we do. So we generate blood every day. Right? That's the most recycled thing that we have. We make order of 10 to 13 cells per day. Huge number of cells being made every day. Right? The question these guys are asking them in macaques, the small bees, figure out where are these cells coming from? Are they all coming from a single stem cell? Or many stem cells that contribute? Right? So each cut here tells you what's the contribution of each cell. Messy. Right? What am I going to get out of it, if at all? Okay, something is dying, some go up and stays, all sorts of dynamics are happening. Very crazy. What happens if you take some time, say tiny, months after that start, and then get the time and ask, I'm not into knowing how much one cell does. This doesn't count. I'm interested in asking, like, when people distribute, like, ask a density function, think of color, or in a language, you want to ask how things distribute. Like, how many cells giving contribute bigger than some? Make sense? Right? You forget the colors, you ask just how many. This is remarkably consistent. So all these months, it doesn't matter which month you look at, the distribution is almost identical. Now there is something to understand, right? Now I want to write down a model where I can understand the shape, where it's coming from, which parameters it may depend on. Of understanding this will still not tell me everything about it, right? There's a certain aspect I've understood very well, right? Clearly from month 2 to month C7, the clones that are contributing the most are very different. Right? Blue clone that's dominant here, there's a red clone that's dominant here, right? Here, there's 16 pieces, if you will. Right? They all are running down this distribution. The other crazy thing is, you kind of squint your eyes a little bit. Yeah, you can get it here, I guess. Top clones, which are plotted here, I guess. So some of the top clones actually make up like 5 to 10% of the blood. Right? Imagine 10% of all your blood is from a single cell. It's somewhere in your bone. It's insane, right? And there's a beautiful experiment done on a very old person, and this one was 105 something, by tracking the genetic mutations they had in the blood. The illusion was that all the blood this person had came from 1 or 2 cells. Right? So these kind of insights you can actually generate when you start looking at things.
That one when stem cell trees can be affected. Like you get some DO clones there.
Absolutely.
They don't have any.
Absolutely. Yeah. So what I want to say is that, as in physics, we want to think of, there is something in how to look at biology, which can simplify it, how you can make progress in understanding. What you understand is different now. So that's an example of what we try to do when we try to approach logical problems from a quantitative side. Of course, the data has to be good. Someone has to make something quantitative, hard. It doesn't take away anything from this blue track. Love, if anything, right? Some monkeys were tracked for 10 years, like long experiment, multiple monkeys, generating kind of plots. So hard experiment, very hard experiment, But they were asking better questions than what we could. We'll see what we get. But I would. So this idea of structure function, how related when I see something biological. And the structure is something that we are good as engineers. Look at structure of things. We're building something, or we are looking, atom, I don't know, code. We're looking at the structure of these things and figure out how does the structure. But it gives you one more thing, which we usually for dynamics is the function. A piece of rock doesn't have a function. Right? It's there, but in biology, the idea of function also becomes very central. Right? All the examples we were seeing things happen. Right? First cell is trying to hunt something down. You can ask how to do so well. Right? You can develop the brain question, what is sort of all synchronous so well? Does it really need to sync that well? Is it all like that every other organism you see? Solution? All personal, right? And civil. So it's sort of the cliff. This is the way I usually my kind of work, right? We work on principles. This thing we work now. If you go think of genome scale, right? So this in yeast, you have their mitochondria. You know mitochondria? Mitochondria also has one genome. Right? So our cells have 2 types of genomes. One is in the nucleus, the rest is found in the mitochondria. It turns out mitochondria genome like a Frankenstein. They kind of recombine a lot and make these weird structures, right? So it's kind of measuring structure of. You can see this makes beautiful repeated structure again and again. It's not the real mitochondria looks like. This is a non-functional structure. So what kind of structure is created? Second, you ask, well, a lot of work thinking about microbes. If you think about bacteria, we only think about bacteria. Right? Bacteria are affected by viruses. Right? We only remember, invented by viruses. Bacteria has to really have this every day. In fact, the numbers are massive. So much of stage bacteria interact in pods. Right? This was really discovered. So what's the caricature of the food chain? The food chain is there's little bacteria at the bottom in ocean. They use light finance, right? Photocyanobacteria, which kind of use light and fix carbon, nitrogen, phosphorus into something, organic matter. Other bacteria eat it. These are organic, these guys are made in some ways. These are eaten by, the grazers come and eat bacteria, and the fish come and eat that, and then we eat fish, right? And so this food is how the food comes up in the system. Now, what's happening right in the middle of the food chain, very early on, is efficiency of the food chain is actually pretty good. We also see 70% of the bacteria are killed by the virus. So the carbon with bacteria kills like carbon because the virus is killer. It's the same called the viral shunt. The shunt is kind of shunting off of what this chain should look like. Right? But what is miraculous is because it's phenomenon of now the evolution can really operate. Bacteria is going to develop mechanisms so it can evade the virus. The virus is going to discover mechanisms to infect it. So this is a fantastic laboratory where new mechanisms are constantly getting evolved. CRISPR system, famously about gene editing, discovered by bacteria. Discovered bacteria, and then spoke it. It's one example. Everything you use, ERs, this, or if you're doing animal genetics, all the tools. Okay. This is a great laboratory. This is happening every second, every millisecond, and the event happen, right? So this evolutionary arm, if you will, kind of has provided a great, laboratory to generate very instant. So we studied this idea of actually with CRISPR. So what a bacteria does, it actually discovered a way of immunization. If a phage comes and a virus comes back, it keeps a little memory that in some cell. If a few cells, a few memories, 20 memories, and the same bug comes and acts again. It's like the vaccine, right? The same dynamics, right? It's a vaccine itself. Or when we get a flu, we haven't got, but we get a flu. Our system remembers. T cell cells remember what happens. That's why mostly we don't twice in a season, unlike mine. But most times we get flus. Right? And for the next season, because it goes together, we get a bunch, comes back, and the time our memory is kind of wiped out. So similarly, these guys work. Right? Might have a memory, the virus is going away, and I know different solutions. You might get this memory. It becomes like an untrace learning in memory. And what do you remember? You forget. You can those dynamics. And it's very similar to what was happening in COVID. It's very similar now. Viruses mutating ourselves, vaccinating ourselves. This untrace is precisely thing happens. So I think these also help us a very good sense of what might happen in us, for instance. Yeah. It's a good question. So in many systems, one or the other might be laboratory. It's not surprising one or the other, right? So that is a whole host of questions. Well, how will the system even manage to fly that long with both being on? Right? We don't have an answer. In fact, you go and let a dirty soil or gut or many species right together. And she said, well, if Darwin was true, Darwinian dynamics true, one of them taken over. Right? Why complexity casts itself? Those are kind of questions which are not answered entirely. You figure out what are the measures of how we can together, in gene together, what genes might fit, and that's the question we can do. It's a very dynamic type of. You have to know biology as well. And this is one question that you had, right? There's some questions, right? We'll get in a few minutes, right? It's a bit long. And then most recently, thinking about stem cell development. Let's see how. So the question is, so what? What you can understand? That's fantastic. I mean, reason of studying conductor physics or semiconductors is being able to make our own. To practice, maybe elevated brains, some might want to understand. How that is really the way of all of it. No, you ever ask that, why are you studying this? It's just so fascinated by it works. And I think that biology is like that too. It's a fascinating thing. You just want to understand. And I'll do it. But let's also give you a sense of that is a what as well, if you want to what exists. And some of you have thought about it, For example, you talk about stem cells. You grow these cells in wax. Like a lot of them are you want to do a third. You grow these cells, same dynamic one of some of the mutants. Right? When this you don't want them, those ones growing faster than the other ones, it's actually not functional. Right? So you can find a way of kind of some way thing allows you to do that. About tumors, the cancer, right? One of the things is able to diagnose. Even if you treat them, relapse. So you can be thinking about when, how that is, but that's okay. I was thinking about thinking, starting dynamical models, starting about predictions, right? We think in very physical models. No idea to biology. You can use machine ideas, you trying to use these are things that you're trying to learn and you don't know how to write in this system. You kind of neural networks. But you data. It's what I think biology is now, which is allowing people to actually make a lot of progress. It's very high quality, good data coming up, much more. So kind of shift paradigm even. Used to be in a place where the data was scarce, very little. You have to be this very model and very little data predict. Now it's actually opposite. Data is coming up as a tsunami, if you right. We don't have frameworks, mathematical or conceptual infinitives. Right? So people who are just kind of analysis developing it's a great time to do that. And then the communism, you can get a trans, right? And that's all the problems. Right? But how happening? We do that. And I think time. Let me go down after 5 years. My goal was to give it more. Right? It's going to be a good place to start taking rather than me talking. Alpha 4, you must be. Yeah. Yeah, I mean, I won't have time to describe it. Imagine what's happening to a stem cell. Your bone marrow. That stem cell didn't rise to a blood cell macrophage, all these different cell types, right? But what does this mean? This means that all these, the stem cell expresses a very different set of genes than what my eye cell expresses, for example, right? Or a blood cell. It's sort of going around, in oxygen from the other, or a blood cell. It's different from a stem cell where it came from. Are you teaching? Oh, okay, I'll give you a minute. Right? So the question is, how do you know paths these things take from right? And we can do a lot of things, but we don't have a good model to do that, right? So we can use, so the idea was, you measure a lot of these gene expressions, you know which genes are getting expressed. If you want to put them in apps, so here they use actually these neural OEs, which is a very machine idea, to kind of find out how cells, which paths will actually when they go from type to type. Use metal data. Right? So you have measured these things, you need to write down analysis to take from one to this way you learn problem. Uses similar idea, more dynamics. Maybe question if you want. I'll change this at the end. Yeah. You could, yeah, you could think of the density chasing some image. You just say you're the thing they want to talk about. So you can think of it as way of doing it. That could be one. But you need some gradients. Right? Oh, yeah. Right. Good cooking.
Thank you very much.
Of course.
That's fascinating.
Right. Yeah, so don't hesitate to ask questions. Also, if you guys are interested, lots of biophysics students here in the department, and you should reach out to them and talk to them. I think they are the best sort of people to really get into understand what they're doing, why they're doing, things like that. And I'm happy to put more people in touch with them as well. If you can get. Yeah.
We'll post the talk in the PhySU.
Perfect.